I'm excited to guide you through a virtual tour today. What you see on the cover page is an image of poplar growing inside the Apple facility. Apple is a unique facility. It's designed to characterize performance of plants both physiologically and metabolically through a series of detectors, sensors, and cameras. And we'll dive into the details of all of that. Let's go ahead and switch to the first perspective on the virtual tour. And I wanted to introduce Kim Askey, who is actually driving the video tour. So you'll hear me reach out to Kim. Kim, can you show us this? Or Kim, can you turn to that direction? She's behind the scenes, and I really appreciate her being willing to guide us through the Apple tour. And this is what's called the buffer or the cultivation area. We've targeted poplar, as you see in the image here, and switchgrass. And those are our two main feedstocks. Through a series of fermentations and catalytic upgrading, we can now produce a full suite of hydrocarbons used in aviation fuel. 50% of the current demand for aviation fuel represents about 20 million acres of agricultural land. So we're looking at not displacing food, not displacing corn or soybeans, but capturing marginal economically available land that experiences periodic drought or has soil nutrition issues. And so we're using Apple to characterize poplar and switchgrass water use efficiency, nutrient use efficiency, and so on. And we'll dive into some of those issues here in just a second. Well, if we capture 20 million acres of land for the production of aviation fuel, it represents about 10,000 new rural jobs, and it represents domestic energy security, something that we would all strive for here in the near future. So let's go ahead and talk about what we're looking at. If you look at the cursor that Kim is moving, you see those beige boxes on the ground, and those are fertilizer and watering stations, what we call fertigation. They're controlled by a series of computer commands triggered by QR codes on each of the trays, the blue trays that you see to the left. So as an individual plant moves down the conveyor belt, scan reads the QR code, and the computer determines whether the plant should be watered or fertilized or weighed, or if we're to inoculate it with a microbiome, a series of bacteria and fungi that may convey favorable plant performance. You'll notice that there's green targets throughout the entire image, and those launch videos. So Kim, let's go ahead and launch the first video, if you will. What we're seeing here is a plant moving up the conveyor belt. And so this is a conveyor system. You can see the QR code on the front of the tray. And as the plant moves up, it turns a corner and moves into the fertigation station. And so each day we can measure, water, and move up to 500 poplar plants. So the image that you see right now is the buffer or the cultivation area full of 500 alternate genotypes of poplar. You'll see some are tall and some are short. Some have a lot of leaves, some have very few leaves. And Kim, if you want to launch the aerial view, we'll get an idea of kind of the scope and scale of how different these plants are. So the plants in the video we're looking at right now are about 2 or 3 weeks old. So they're relatively short, about 6 inches tall. Each of them has been sequenced. We know the genotype, we know the variation at the DNA level of every gene and every genotype in the buffer. And with that information and the data that Apple provides, we're able to link drought tolerance or water use efficiency to specific genes controlling those traits. Kim is showing us a view of the light system. So growing plants inside a greenhouse requires supplying light. And we use LEDs to supply 100% of sunlight. So there's about 2,000 micro-Einsteins per meter squared of light delivered to the plants in the buffer. That allows us to evaluate or judge maximum productivity. Kim, let's move to the back of the buffer. So we're going to change perspectives here. And we're now looking up towards the area we were standing in. Again, there's a series of targets, and I forgot to mention on the opening slide, there's a QR code. We'll see it at the end of the presentation, where you can enter into the virtual tour and move around, click on the targets as you are so inclined, and get an idea of the scope and scale of how this works. What you can see from this image is that there's a lot of variation in the plant systems. So we had to design APPL, create a combination of sensors, detectors, and cameras that would fully capture performance of individual plants. If we stop right here, right now, the plant to the bottom right is very short. The plant behind it is very tall. And so the APPL system had to be designed to obtain information on plant performance in a uniform environment. And we'll talk a little bit more about how we do that in just a second. As Kim pans further to the left, we can see the conveyor belt entering into what's called the image capture array. Let's go ahead and move to that perspective, Kim. So now we're looking back towards where we were standing a moment ago, and you see the series of conveyor belts. Plants would move down the conveyor belt to the center of the image and into the image capturing chamber. We have a series of different cameras, detectors, and sensors. The first station is a 3D LiDAR scan of every plant, and we'll show some examples of that in a minute. But the 3D LiDAR recreates a point cloud of every plant. This is important because as the plants move through the image capturing tunnels that we see in front of us right now, cameras are moved in proximity to each plant. We know that the information from a subject to the camera varies by distance. So we try to design the system so that we have the exact same information for every plant. So the 3D camera captures the size and width of the plant. Then it enters a light adaptation chamber, which we're seeing here on the video. And their plants are in complete darkness or complete light just prior to moving into the chlorophyll fluorescence chamber. You saw the light triggered and the plant image is captured. From there, it moves out of the back of the chamber, around the back, and into the thermal image station. From the thermal image station, it moves into the multispectral and hyperspectral stations. We'll show examples of those in just a minute. We use the multispectral system as a discovery platform. So let's go ahead and show some examples of what some of the images look like. So this is a 3D image of a poplar plant. Once the image is captured, you can rotate that plant in 3 dimensions. This gives us the opportunity to determine the plant height, to determine the leaf size, leaf number, and the canopy size of the plant. And those are all predictive measures of performance. The nice thing about APPL is that we have accompanying all the APPL data, years and years of phenotypic data or trait measurements out in the field. And so for each genotype that passes through APPL, we know its performance at multiple sites under alternate conditions. But APPL gives us the opportunity to do time series. In a field campaign, we may send 5 or 6 people out to measure 3,000 trees over a week's period. And we have that data, and we can use that data to link traits to genes. But in the case of APPL, we measure each plant every day. And so you develop a time series understanding of how metabolism changes, how physiology changes, how anatomy changes. And this gives us insights into different genotypes and their response to stress, to pathogen attack, or to favorable conditions. And with that, we can then predict performance out in the field. So from here, we go into the chlorophyll fluorescence chamber. Let's go ahead and change to the next image, Kim. The image you see on the left side is an RGB red, green, blue image, what we see with our eyes. And you can see the outline of the canopy, individual leaves. You can see differences in color. But on the right side is the chlorophyll fluorescence depiction. And chlorophyll fluorescence measures the amount of CO2 that's being captured by individual plants. Plants have a structure on their leaves called stomates, and stomates are just microscopic pores on the surface of leaves, all plants, that allow CO2 to enter into the plant. And through the process of photosynthesis, CO2 and water are combined to create glucose. And glucose is the building block of all biomass, whether it's cellulose in the form of structure or it's a metabolite in the cytoplasm of the plant. What the chlorophyll fluorescence images allow us to do is pinpoint differences in photosynthetic capacity. And so on this particular image, we see that not all leaves are photosynthetically active, and not all leaves are equally photosynthetically active. And so with this information, we can identify the genes that control the rate at which photosynthesis increases over a period of time. Let's go to the next image, Kim. This is the thermal image. On the left-hand side, you see what the plant looks like to you or I with our eyes. But on the right-hand side is the thermal camera. I mentioned earlier that plants have pores or stomates that allow CO2 to enter. But those pores are also liberating water, and that water is a mechanism for cooling the plant. If the leaf temperature gets above 40 degrees C, photosynthesis shuts down. So the plant regulates its leaf temperature by liberating water. But if a plant is under water stress, limited water availability, those stomates close. And once the stomates close, leaf temperatures elevate. And so we can use the thermal camera that we see on the right to depict variation in leaf temperature and therefore variation in photosynthesis, the ability of the plant to withstand limited water and still remain productive. I mentioned earlier that in CBI, we want to capture marginal agricultural land, mostly in the Western Great Plains that experience episodic drought. And so we want to identify genotypes, individual plants, and genes that then allow that plant to grow under limited water. And we can use the thermal camera here to identify those individuals and then ultimately those genes that'll allow plants to be more productive under limited water availability. Let's go to the next slide, Kim. This is a composite of a single plant out of the hyperspectral chamber. If we were to look at this plant, we would see green leaves. But because we have the ability to measure color, if you will, in the hyperspectral range, we are able to detect up to 500 different colors, not just RGB, but 500 different wavelengths of light. These wavelengths of light can be then painted on top of the point cloud that I mentioned earlier. And what we see here are differences in spectra. So in certain wavelengths, the plant looks uniform, like in the upper left-hand corner. That plant at that spectra looks fairly uniform. But as we move from alternate spectra across the entire range from 350 nanometers up to 1,750 nanometers, we begin to detect hidden variation in the reflectivity of the leaf to the camera. We're using this platform for discovery. So we would like to develop through AI and ML approaches, predictive algorithms that can be then used to indicate early onset of drought stress or early onset of nutrient deficiencies or pest attacks. We can then take this information, design a camera with only those spectra that are informative instead of all 2,000 spectra. We can then design this camera small enough to fit on an unmanned aerial vehicle. that can be deployed daily by landowners who are producing biomass for energy. So for example, if you were a landowner in Western Nebraska and you had a switchgrass field that you were hoping to sell for bioenergy purposes, the drone would automatically survey the field daily and indicate when a water stress event is about to happen, not after it happened, but is on the verge of happening, or a nutrient stress event, or the occurrence of a pathogen. And then that allows the landowner then to intervene and correct the conditions under which the plants are grown. And so we have a team of computational scientists working very closely with our computer science directorate, and we've used Summit in the past. We hope to use Frontier in the future to build these predictive algorithms, not just on the drought tolerance, but on productivity, on cell wall chemistry, on, of course, insect and disease resistance. Let's go to the next slide, please. So I wanted to highlight one example. What you're looking at here are poplar grown over a 90-day period. Baruch and Wellington are about 5'10", 5'11". So it gives you an idea of how quickly poplar can grow over a very short period of time. The tree on the center, the tall tree, is the exact same genotype as the tree on the right, except for one gene. And we discovered a gene called booster that improves productivity. And you can see that with your naked eye. You don't need fancy cameras or detectors to know that that plant is bigger. But how we detected that gene was using chlorophyll for fluorescence. And I mentioned earlier that in the process of capturing sunlight and CO2 combined with water, you produce glucose. But each genotype's rate at which they reach the maximum photosynthetic capacity varies. So some genotypes under low light elevate to the maximum photosynthesis in a very slow manner. Others hit that maximum and have an elevated maximum that allows them to create more glucose. The more glucose there is, the more cellulose there is, the more hemicellulose and lignin, which are the three major components of the cell wall. So by identifying this gene, isolating that gene from an individual, and inserting it into the genotype on the left, we were able to demonstrate about a 200% increase in productivity. Now, this gene, interestingly, is a chimera, or a combination of a motif, a DNA motif from a bacteria, a DNA motif from an ant, and a DNA motif from rubisco, the protein associated with photosynthesis. We've cloned that gene and we've placed it into rice, Arabidopsis, tobacco, and we see similar increases in productivity. This gene, we're now in negotiations with several companies to license this gene for applications beyond bioenergy, applications to food production or forage production. And so this is our first example, although Apple's only been operating for about a year, fully commissioned. This is the first example of successful identification of a gene that will improve the rural economy, ultimately leading to the development of sustainable aviation fuel. Apple generates, if we were to maximize the number of plants and have plants in Apple every day and be measuring all 5 modalities for all plants, we would generate up to 7 petabytes of data per year. And so we're in combination with computational sciences, looking at developing edge computing capabilities here in Apple. So instead of just being automated, we would like Apple to be autonomous. In other words, make decisions about nutrients and fertilizer applications, or make decisions about water applications. That isn't in place yet, but we're looking down the road to the future where we'll have an autonomous greenhouse generating data for the bioeconomy. Before we leave this image, I wanted to say we're still adding new modalities to Apple. We're adding a new below ground imaging system that should be installed in May. With that, we'll be able to not only image above ground portions of the plant, but we'll be able to image the below ground portions. This complements the work at HIFER and SNS, where we use neutrons to evaluate root invasion, water content in soils. We hope to unite images from SNS and from APPL and draw insights, biological insights into performance. I want to highlight the QR code on the bottom left. This will bring you into the APPL tour. You can move around, pivot, turn, elevate, or click on any of the green targets in any of the positions that I've highlighted. I would encourage you to do that. Go ahead and share the APPL QR code with your kids when you get home. Share it with your neighbors and friends. And let me just close by saying thank you for your time. If you're interested in APPL and you think you have an application that could benefit from APPL's skill set, please contact me and we can set up an opportunity to discuss future experiments.